All right, so good afternoon, everyone. Today I will share my work on multi-data learning, and more specifically, I will introduce you to a multitude of ABCs. So nowadays, and in a numerous amount of projects, data is being collected in huge amounts. And we can actually regard, well, this data is often stored in multiple data sets and in a variety of types. We can actually regard these data sets as different pieces of a puzzle that we are trying to solve. We are trying to solve the research question, and we have multiple data sets available for this to do this. Now, in order to solve the puzzle, we will perform an analysis. And an analysis often consists out of communication with an internal team, but also with the experts and the scientists who have collected the data. It also consists out of research, coding, efficient programming, database storages, and often such a loop is interpolated multiple times before eventually the pieces of the puzzle fit together and we can provide an answer to the question. Now, an example or an illustration of a project with multiple pieces or multiple data sets can be found in drug discovery. So here, one of the most prominent goals is to try to discover the mechanism of action of the compound. We would like to know how the compound is behaving inside the cells, and by this we would not only like to know the desirable effects, but also the undesirable effects. And it's actually a multidisciplinary field. So we have chemistry, because we have to know what the compound looks like. We have to know how the nicks and the necks of the molecule are before the key can fit the lock. We have proteomics, because the compound has to hit the target. We have genomics, because we have to measure gene expressions. And we also have system biology, because genes are part of a pathway analysis. Now, all of these data sets are actually interconnected. We will see a certain change in a cell because a pathway was activated, and it was activated because a gene was being regulated differently, and the gene was activated because the protein hit the target, and it did so because it had a very specific formation of the molecules. So we would actually like to combine the information from all of these data sets together. Now, we could actually say that a single data source is quite limited in its point of view. If you are looking at the data set 1 and you will perform a cluster analysis, the compounds or the objects that you will find similar to each other are going to express this similarity only on the first data set. If, again, you have a second data set and you perform again a clustering analysis, you will find objects that are similar, but they are not necessarily similar on data set 1 because you have not used the information that was embedded in data set 1. So therefore, we can actually try to extend our knowledge by integrating multiple sources of information and maybe even perform clustering on both datasets at the same time, such that the objects that you find in clusters will express similarity not only on dataset 1, but both on dataset 1 as well as on dataset 2. And to this end, multi-source clustering techniques have been introduced already some time ago, but also recent techniques are being introduced. So what would you like to gain from such a method? There are 3 objectives which we would like to see in the end result. Such an ideal integrative clustering technique would like to discover, well, first, similarity that is consistent across the multiple data types. If I find objects that are similar on dataset 1 as well as on dataset 2, I would like, of course, to see this in the end result as well. But we can also not forget the similarity that was very specific to a particular data set. Suppose we have objects similar in dataset 1, they're not similar in dataset 2, still it would be interesting to find these objects. And as a third objective, we would like to see objects that are expressing only weak similarity to each other, but on each of the data sets, so that actually the combination of this evidence can still show up in the end result that, okay, the similarity they express on each data set might rather be weak, but in the end, we would still like to see them because it's consistent, it's always there. And for this, I will introduce you to the MABC method, which is actually a multitude of ABCs or multiple aggregation of bundles of clusters. So shortly, what we can say is that suppose that we have multiple data sets, and then for each data set, we will perform R iterations. And every iteration for every data set, we will take a random subset of both the samples as well as the features we would like to cluster on. So we take this subset and then we will perform clustering. And on this end result, we will cut it in a specific number of clusters. And for every result, we will set up an incidence matrix. And such an incidence matrix is a squared matrix, which is binary. The value will be 0 if objects I and j belong to different clusters, so they're not similar. And it will be 1 if objects I and j belong to the different cluster, they're similar. So we will end up for every data set with R of these incidence matrices. So we have R times L, I put in here L, incidence matrices. And what we will do now is we will just sum them. Next, we will divide this summed incidence matrix by the number of times two objects were selected simultaneously to convert it in some sort of a metric. And on this final distance matrix or similarity matrix, we will perform a final clustering procedure. And that will be the end result. Now, the benefit or the advantage of such a method is that the datasets can be of any type, because the first clustering, the R iterations, will be performed data set per dataset. So you can have a continuous dataset as well as a binary dataset as a dataset with all kinds of information. I'll give an example of that later on. And any clustering algorithm can be applied. So if you're a fan of a hierarchical clustering algorithm, that can be done. If you would like to use k-means, also not an issue.
Do you have one incident matrix per data set or one global incident matrix?
One incidence matrix per data set per iteration.
And then you assume equal number of nodes, or is that maybe?
Yes, yes. Yeah. So I've created an R package called inclust. A quick overview. Don't be scared. It contains a lot of multi-source clustering procedures. I will not go into detail about all of this. It's just to show that there are a lot of them available. Maybe point out similarity network fusion, SNF, which is a very nice one. But there are also more agglomerative features as well as our own MABC implemented, as well as hierarchy-based clustering approaches. I'm not going into detail, but just to show you that many of them are available already in literature and now also in R. Time for some case study to make everything very clear. So in the first case study, we will be looking at the MCF7 cell line. We will have 56 compounds, which we have two data sets. And these 56 compounds, we have records about their molecular features, which is a binary data set, 0 if the feature is not present, or 1 if the feature is present. And we also have a target prediction data set with, again, the same meaning, of course, if it is a 1, then the compounds will hit that target, and if it's a 0, it will not hit it. So we would like to now combine the information that is present in these data sets, but it's always a good idea to also look at the single source clustering results just to see, to have more familiarity with the data. So we can perform clustering and on just the molecular features, for example, and see what it looks like. And you will end up with a dendrogram which it shows, I cut it into 7 clusters here. Every cluster has given a color. So compounds in the same cluster are showing a similarity on molecular features. So for example, we have here a yellow cluster. A little bit further, we have a blue cluster, which is rather small. But okay, this is what it looks like on molecular features. We can do the same for target predictions. It looks a bit different, but that's basically all I see at this moment. I cannot already connected to the previous picture. So we have come up with a method to compare both results quite nicely. And we will also immediately include the results from our multi-source clustering results. So, okay, let's take a look. Here we see all our compounds. These are the compounds. Here are the methods that we are looking at. So here we have our single source clustering methods, molecular feature clustering, target prediction clustering, and a selection of multi-source clustering results. What does it imply? It means that here, this brown color implies that these compounds were part of that particular cluster. If we check at the next line, which represents the target prediction clustering, we see that this compound has moved to the green cluster, while this compound was put back into the brown cluster. So you can see how the clusters change according to these methods. Now, for example, here we see that these pink compounds are always together, both on molecular features as well as on target predictions. while, for example, here, this yellow cluster, we see they're very much together on molecular features, but under the influence of the target predictions, we see that these two are, changed to the blue cluster. And is this really true? We see also this change, of course, in our MABC method here. Now, of course, the question is this true? Let's take a look. Do my blue compounds or yellow compounds here indeed express this similarity to the blue cluster also on molecular features or not? Let's take a look. So here we see the determination of some characteristic features. Here we have the cluster, we have the 4 blue compounds as well as with the 2 yellow compounds, and in molecular features I can indeed see that they are expressing some similarity. And here we are looking at the pink cluster, which was indeed also expressing similarity on molecular features. So although these compounds were not part of the blue cluster under the molecular feature clustering, they were found to be similar to them based on the multi-source clustering procedures. And also in target predictions, we can see similarities between our 2 clusters. So for both data sets, the similarity has come forward in the multi-source clustering procedures, and we found clusters that are exhibiting compounds that are similar on all features or all data sets that we used as inputs. If you want, you can also include gene expression to test whether these compounds are now behaving differently in gene expression versus the other methods because they are expressing similarity. We might also expect that they are expressing similarity on gene expression as well. Now, in the second case study, we would like to investigate the all leukemia data set, which has 128 leukemia patients, as well as gene expression from over 12,000 genes, so it's a bit bigger than the previous data sets, and we also have clinical data. This clinical data is all about tumor characteristics as well as age about the patients. And we actually know that these patients are already divided in B leukemia types and T leukemia types. The question is, can our multi-source clustering procedures refine these cluster compositions? Let's take a look at this. So here again, we see our single source clustering results. Well, actually, the first one is representing the truth. So this is the true composition, with the blue ones being the blue group, being the B-type leukemia, and the yellow ones being the T-type leukemia. We can see that in gene expression as well as in clinical data, this is not found. But we see here that the multi-source clustering procedures, and especially our ABC method, is indeed refining all of, doing a quite good job of refining the cluster, the leukemia types as they were actually found by the physicians, by the other determinations and other tests. Now, putting it in misclassification errors, we can see that our MABC method is quite good in trying to refine these 2 types of leukemia, just based on the gene expression as well as clinical data, while each individual source is not very good. Now, looking into the result of the MABC a bit better, we can actually see that there are 3 main clusters, and that our B group has been split in 2. So we see a purple part of the B group here, as well as the blue part here. Now, of course, the question is, why did MABC do this? What's the reasoning behind splitting up the B in 2 different B subgroups? And we can see that if we look at the clinical data, that this subgroup of B group 1 is quite specific. So the cytogenetics cannot be normal. There cannot be a T411 translocation, and there must be a T922 translocation. So it is really a specific group of B-type leukemia that the MABC has found. And also in gene expression, we can see a difference between the 2 B types in our data. So some concluding remarks is that integration of multiple data sources is very beneficial because we are looking at multiple aspects of the underlying biology, and we can actually try to save resources by exploring the mechanism of action in silico. So we can see that ABC is quite a promising multi-source clustering method, as we can find strong similarity as well as individual similarity, and simulation studies have shown that also if weak similarity is shared, it shows up in MABC. So the R package is unclassed. If you would like to explore some other methods, it's available on cron as well. And then all that's rest is to thank you for your attention. If there are any questions, I'll be happy to answer them.
So thank you for the presentation. So we have a few minutes for questions.
Thank you for your presentation. My question is about the number of clusters.
Yes.
What is the method that you use to get that number?
For this particular dataset, we use the gap statistic, which is also available in the R package. If you want to perform clustering, you can just put gap to true, and then it will determine an optimal number of clusters based on the gap statistic. But of course, there's discussion about the optimal number of clusters, and I think multiple methods are out there to determine the optimal cluster.
I do have a related question. How do you know that in the different data sets you have the same number of clusters?
Yeah, that's you don't, of course, for really know, but it's a bit of an exploration. You can try out different number of clusters and then see which one fits your result the best. But there are some methods, and I think it's the hierarchical clustering, HEC, that determines a bit their own optimal number of clusters. So you give it the data, and then it will give you an optimal number of clusters according to its method. Because that's one of the most trickier things to do, of course, if the number of clusters differ.
Have you considered using cluster methods that have better way of model choice regarding number of clusters than gap statistics? So there's some model-based clustering methods which can provide AIC, BIC criteria, and there's also something for non-parametric clustering.
I haven't tried it so far, but it sounds quite interesting to explore further and to maybe add to the package as well. Okay, thank you.